Hello, good morning, everyone. Very nice to have you here for this third session. We are going to have 4 technical presentations today. We have 3 full papers and a short paper. And at this point of the event, I think that you already know how the sessions are working. We are going to listen to the 4 presenters, and you can feel free to write down your questions on the YouTube chat here. And in the end of the session, I'm going to pick some of the questions or all of them, if you are lucky. And I'm going to call all the presenters back, and we can do the questions part, okay? So I'm going to call our first presenter, Felipe Vaz Perez, who is going to present his work entitled ContFree-NGS, Removing Reads from Contaminating Organisms in Next-Generation Sequencing Data. Okay, Felipe, the floor is yours. You can share. Yeah, it's here. So I'm going to put the video, and let's go.
Thanks.
Hi everyone, my name is Felipe Vaz Perez. I am undergraduate student in biotechnology, and today I'm gonna talking about ContFree-NGS, a simple software that enables the user to remove contaminated sequences from their next generation sequencing dataset. So to start this presentation, I would like to introduce the problem of contamination in public datasets. So, as defined by NCBI, a contaminating sequence is one that does not faithfully represent the genetic information from the biological source organism because it contains one or more sequence segments of foreign origin, and they could cause several problems in downstream analysis. The primary consequences of contamination are time and effort wasted on meaningless analysis, erroneous conclusions drawn about the biological significance of the sequence, disassembly of sequence contigs and false sequence clustering, delay in the release of the sequence in public databases, and the pollution of public databases. So, ContFree-NGS was developed to solve a problem I faced when I was working with sugarcane.
Seems that Felipe left the session. Maybe he had some problem. I suggest that we should call the second presenter and wait for Philippe. Maybe he's going to come back. So, Gabriel, are you ready? Can I call you right now? Okay. So, I'm going to call Gabriel. He's going to present his paper entitled Deep Learning Based COVID-19 Diagnostic of Low Quality CT Images. So, welcome, Gabriel. I'm going to show here your presentation. And good luck, good presentation. Thank you.
Thank you. Uh, so I am Gabriel. I'm going to present the paper Deep Learning-Based COVID-19 Diagnostics of Low-Quality CT Images. The authors of the paper are myself, Daniel Ferber, Felipe Vieira, João Dalben, Mariana Ferraz, Nicolas Sato, Rafael Padilha, and Zanoni Dias. This is the agenda of the presentation, so I'm starting with the introduction, then I'm discuss about the dataset, methodology, experimental evaluation, and conclusions and future work. So the COVID-19 has impacted healthcare systems and economy activity and citizen lives around the world. And to make the diagnostics of the COVID-19, it's necessary to use, for example, PCR tests, and this kind of test is time consuming and it require a lot of effort. With that, many students and professionals in the community started to use machine learning and deep learning classifiers to help the prediction of COVID-19. Uh, these kind of classifiers use blood cell counts, chest X-ray images, and computed tomography scans for training these classifiers to make the prediction. Considering the CT scans, it presents a higher sensitivity than X-ray images for this problem and have been successfully used in many diagnostics of diseases using computer programs. However, this kind of data has some problems. For example, the different conditions of sensors of different machines depose that the patient are put in the bed and the clothes that the patient are using. So with that, there are many problems of the different images in the CT scans from different hospitals or machines in the exams. So with that, in this work, we evaluate several preprocessing techniques to reduce the impact of low-quality CT scans using deep learning-based diagnostic tools for COVID-19. So in this problem, in this article, we use the COVID-X CT dataset. So this dataset has images from patients, CT images from patients, and each patient has different images from the same exam. So one exam is composed by slices from the lungs and chests of the patients. And this dataset has three classes. The first one is the normal, which indicates that the patient has no disease. The second one is the common pneumonia, and the third one is the novel coronavirus pneumonia or COVID-19. And with that, we can make the prediction considering the image level, predicting the slices per slices of the patient, and considering the exam level with the whole slices of the same patient. This dataset has images from different hospitals in China. So with that, we can see different aspects of each image. For example, there are images with rounded box, for example, in the letter A. And we can see too that are some problems with the clothes that the patient are using. The structures of the bed that the patient made the exam. Some problems with reflections in the chest area. And here in the letter A, we can see that there are problems in the background with a noisy in the background. So The model need to see the chest area of the patient and not to focus on the background or some aspects in the image background. So with that, we make our methodology to make the classification considering these different kinds of imaging and aspects of the image. So this is our pipeline of our methodology from the exam level, exam of a patient. We made some image processing techniques to create a new images, preprocessed images without any kind of noise in the background. And with that, the model can learn to make the diagnostics considering this image. So with this processed image, we make the classification using convolutional neural networks. And here we consider two kinds of level diagnosis. The first one is considering the image level diagnosis. That's it. We can make the prediction considering only one slice for one patient. And with that, then we can also make the exam level diagnosis considering all the slices of the same patient to make the final prediction of the exam. And in both of them, we make the classification considering these three classes, normal, common pneumonia, and COVID-19. So in the first step, in the data processing, we use this pipeline to make the filters of the image. So from the original image, we apply a 5x5 kernel to reduce the noise using a minimum filter. Then we make the binarization and edge detection to make the segmentation of the image. From the detection, we apply the background fill to complete the background with black pixels. And then with that, we can create two masks. The first one is the interior chest mask that considers only the lung part of the patient. And the second one is the external chest mask that considering the exterior chest part of the patient. And with that, we apply these masks on the original image. From this, we can create 6 different image representations. So the first one, we apply the exterior chest mask in the original image and make a bounding box of rectangular shape from this output image. Then we have the rectangular center that is similar to rectangular, but in the end we center the output image for this kind of preprocessing technique. We also have the elliptical form. So we apply the exterior chest mask and then apply a elliptical bounding box form to make this image. And equal to rectangular center, elliptical center is the elliptical form, but the image is centered in the output image. And we have also the exterior chest that is only applying the exterior mask on the original image, and the interior chest, that is the application of the interior mask in the original image. So here we can see these preprocessing techniques. So this is the first one is the original image. Then we have the rectangular preprocessing technique, elliptical preprocessing technique, exterior chest, and interior chest. In the case of interior chest, in the background we put all the pixels as white pixels. Then with this kind of image, we need to make the classification. So to make the classification, we use two different convolutional neural networks architectures. The first one is our baseline architecture, and this architecture is composed by 3 convolutional blocks, followed by a block of fully connected layers. So each one of these convolutional blocks has 2 convolutional layers, followed by a max pooling with 2x2 kernel size and a dropout operation with rate 0.2. The number of convolutional filters starts at 22 for the first block and is multiplied by 2 in each subsequent block. And in the fully connected layers, we have 3 layers. The first one has 256 neurons, the second one has 128 neurons, and the last one, the output layer, has 3 neurons to make the classification. And the second architecture that we used is the ResNet 50 architecture. So this is our experiment. So this is the pipeline of our experiments. The first one is using the data processing, the different data processing techniques with the baseline architecture. So we started with the baseline architecture because it has fewer parameters than the ResNet 50, and it can run faster than ResNet 50. So with the two best preprocessing techniques, we apply ResNet 50 with different number of unfrozen layers for this architecture. Then we have the different image resolutions with the best configuration of ResNet 50. And then we make the prediction on the test set. And finally, we make the interpretation of the model decisions using GradCAM. So in the first experiment, we used the baseline architecture with different data processing techniques. So here are the results of our first experiment. There is the different metrics. We use different metrics. The first one, the ACC, is the balanced accuracy. That is a common metric used in the multi-class classification. And the second one is the COVID-19 false negative rate. So it's important to use this metric because when a patient has COVID-19 and the model predicts that this patient has no COVID, it's important to not occur this case because it's very hard for a patient that has the disease and go home. And using these two metrics, we evaluate our preprocessing techniques. So here are the results of different preprocessing. So the best result was achieved by the interior chest that achieved 94.53% of balanced accuracy and 1.31 of COVID-19 false negative rate. And the second best, considering the COVID-19 false negative rate, was the exterior chest, which achieved 2.04% of COVID-19 false negative rate. And we used these two configurations for the next experiment. But it's important to notice that the no processing had the second best balanced accuracy, considering these preprocessing techniques. So with the two configurations, exterior chest and interior chest preprocessing, we applied the ResNet 50 with different number of unfrozen layers. So we used 0, 12, 24, 32, and 40 unfrozen layers. And again, we used the balanced accuracy and the number of false negative rate for COVID-19. And here, the best results, considering the false negative rate of COVID-19, was using 32 layers and exterior chest for ResNet 50. So with that, we made the third experiment. There is a change in the resolution. So until now, we were using 128 per 128 image resolution, and then we applied 224 per 224 resolution. And with this higher resolution, considering the exterior chest and the ResNet with 22 unfrozen layers, our model achieved a higher balanced accuracy and a lower number of false negative rate for COVID-19. So with that, we select this as the best architecture. So in this experiment, we use the validation set to find the best configuration of ResNet 50 and the preprocessing techniques. So Then we apply this best configuration in the test set. So considering the image level diagnosis, our method achieved 97.84% of balanced accuracy, and looking at the confusion matrix of image level diagnosis, we can see here that the false negative rate for COVID-19 is close to 1%, so it's close to our results in the validation set. And then we apply considering the exam level diagnosis, and our method achieved 99.5% of balanced accuracy in the test set. And here in the confusion matrix of exam level, we can see that the false negative rate for COVID-19 is close to 0. Finally, we make the interpretation of the model decisions. For this, we used GradCon. And here are two cases. The first one is the no processing technique, considering the original images. And the second one is the best pre-processing techniques, considering the exterior chest. So here are the three cases. The normal, there is a patient with no disease, novel coronavirus pneumonia or COVID-19, and common pneumonia. And here we can see that with no processing technique, the model in some cases are looking or paying attention on the background. So in the case of new images, the model probably will use some kind of noise in the background to make the classification, and it's important to model to pay attention on the correct part of the image, and it occurs in the exterior chest that we can see that the model is paying attention on the parts of the lungs and the chest of the patient. So with GradCon, we can see that our model with exterior chest is paying attention on the correct part of the image. So as conclusions, we conclude that it's important to remove noise from CT scans and CT scans from different hospitals or machines and sensors. It's very important to make this pre-processing step. And the complexity of the models and high resolution images can improve the results compared to models, simple models and low resolution images. For our future work, we can highlight the use of ensemble techniques to improve the results and the investigation of different convolutional neural networks to make the prediction of CT scans. We also can highlight the investigation of complementary of preprocessing strategies to make another steps for preprocessing the images. And here are the references used in the presentation. Thank you.
Thank you, Gabriel. The public is also congratulating you for your excellent presentation. It was really very nice work. We have questions already, and I ask the public to keep putting the questions on the chat. And in the end of the session, we are going to deliver these questions to you, okay? So now we are going to call the third presenter, in fact. The work is called Feature Importance Analysis of Non-Coding DNA RNA Sequences Based on Machine Learning Approaches. I think that the presenter is Breno, right? Breno, am I right? Okay.
Yes.
Right. So I'm going to call Felipe back after your presentation. Okay. Can you share your presentation with us?
Moment.
Okay. Oh, okay. That's here. Okay. Oh. Can you try to share it again? Because I think that's not working. Yeah, no. It's tricky. Wow, so weird. Well, I think you could try to stop sharing and share it again,
Yes.
right?
Let's do that.
It worked previously. Let's see.
Okay, I'll share again.
Okay.
Moment.
So while you are sharing the presentation, I just would like to remind everyone that tomorrow we are going to have, in the closing session, we are going to review the best paper award. So don't miss it. We wait for you. I think it's 3/4 past noon. I think that's it. No? Maybe you can try to send it for me if you want. I can give
Un
you my
momento,
email.
I'll reload the page from the stream.
Okay. I think that he was shut. Well, what we can do here is no, he's back. If it doesn't work, we can call Felipe back, and I also have Nelly's presentation here, so you can try again. If not, I send you my email, and maybe I can pass the slides for you.
Yes. I think it's better to call Felipe.
Yeah. Okay. No problem.
Yeah.
So, Felipe, are you ready?
Yes,
Okay. So let's try again. Are you going to send your video?
I'm gonna share the video again.
Yeah. Okay.
One moment. Oops, isn't
Wrong video.
wrong video.
Okay.
BSB. Okay.
Right. I think that's it.
Yes.
Okay. Let's try. Good. Have a good presentation.
Okay. Thank you, Raquel. Hi everyone. My name is Felipe Vasperes. I am undergraduate student in biotechnology, and today I'm gonna talking about ContFree-NGS, a simple software that enables the user to remove contaminated sequences from their next generation sequencing dataset. So to start this presentation, I would like to introduce the problem of contamination in public datasets. So as defined by NCBI, a contaminating sequence is one that does not faithfully represent the genetic information from the biological source organism because it contains one or more sequence segments of foreign origin, and they could cause several problems in downstream analysis. The primary consequences of contamination are time and effort wasted on meaningless analysis, erroneous conclusions drawn about the biological significance of the sequence, disassembly of sequence contigs and false sequence clustering, delay in the release of the sequence in public databases, and the pollution of public databases. So, ContFree-NGS was developed to solve a problem I faced when I was working with sugarcane RNA-seq datasets. So, as you can see here, that is the taxonomic information of the contaminating sequences in the sugarcane datasets that I was working with. So here you can see the number of reads and percentage of contamination. So Acinetobacter baumannii is a bacteria, and it have almost 3% of reads in this data set. So I tried to remove these sequences of contaminants with some softwares, and recently some tools have been made available that aim to remove sequences from contaminating organisms in next generation sequencing datasets, such as the Contaminer, a tool to unravel the presence of contaminating sequences in the set of reads that do not map to a reference genome, and also like Contaminator, a software that removes contaminating sequences from contigs, exploiting a taxonomic assignment file, and the QC Blind, an automatic tool to do unsupervised assembly and contig binding to identify and remove putative contaminants. But all of these tools have in common that they either require a reference genome of the source organism or need to perform assembly prior contaminant detection. So our goal with ContFree-NGS was to develop a simpler tool to remove contaminated sequences directly from unassembled reads without mapping and exploiting fast camera analysis implemented in taxonomic assignment engines commonly used in metagenomics, such as Kaiju, a fast and sensitive taxonomic classification software for metagenomics, and Kraken 2, a taxonomic sequence classification system. So here you can see our approach. ContFree-NGS was implemented as a single Python script using the BioPython module and the Python environment for tree exploration. And in order to access the contamination, ContFree-NGS exploits a taxonomic assignment file containing the region identifier and the NCBI taxonomic identifier for every sequence in the dataset. So here we can see the workflow of ContFree-NGS. So the first step is to have a dataset in FASTA or FASTQ format. Then you have to generate a taxonomic classification file, and with this taxonomic classification file, can run ContFree-NGS. The user must insert the target taxon, so all of the descendants of the target taxon will be maintained in the final filtered sequences, but I will talk about these parts later. So here, the first step is the taxonomic classification, and as ContFree-NGS exploits the results from a taxonomic assignment engine, users must use the proper switches to achieve an accurate classification. For instance, a proper value of the confidence switch in Kraken 2. So to perform the taxonomic classification with Kraken 2, we built a custom database containing the following reference libraries. So we have the reference libraries from archaea, bacteria, viral, human, fungi, plant, protozoa, and the NCBI non-redundant nucleotide database. And for example, this is one of the Kraken output. So here in the first column, we have the letter C. That means this read was classified by Kraken. The second column, we have the number, the region identifier. So here is the region 001, and then is the region 002. And in the third column, we have the NCBI taxonomic identifier for this region. So this identifier means that this region is from village plant. So with the taxonomic classification file in hands, the first step of ContFree-NGS is to create a SQL database to store the sequence index to make the contaminant removal process faster. And this is done using the biosec.io index db function from BioPython. And ContFree-NGS was the first software that I developed. So in this step, I tested various ways to do this process faster. And finally, in the contamination removal, ContFree-NGS creates a list with the NCBI identifier for the target taxon and all the identifiers of its descendants according to NCBI taxonomic database. So, for example, in this step, NCBI creates a list with the identifier of the target taxon. So if village plant is the target taxon, that is the ID, and all its descendants, I mean all the descendants of village plant is included in this list. So it so this ID is from sugarcane. Then ContFree-NGS iterates over the taxonomic assignment file, and if the read was not assigned to any taxa, it is saved in a FASTQ file for unclassified reads. You can see it here. And if the read was assigned, it will check if its taxon is found in the list of target taxon descendants. If so, it will save the read to a FASTQ file for filtered reads. Otherwise, the read will be discarded. So this is done here in this step of contamination removal. To evaluate ContFree-NGS, we run ContFree-NGS against 3 sugarcane artificially contaminated datasets. So the dataset A contains 50,000 of reads, dataset 2 contains 250,000 of reads, and the dataset C contains 1,250,000 paired end reads. So in all these datasets, 80% of the reads came from sugarcane, 15% of the reads came from the bacteria Acinetobacter baumannii, and 5% came from the fungus Aspergillus fumigatus. So here is the representation of the taxons in these datasets. And after running Kraken 2 with confidence 0.05, the following sequences was classified in some taxon. And as you can see here, for the 3 datasets, nearly half of all sequences were assigned to a taxon. So we can choose if we will remove these reads or don't. So in the final step of the evaluation, we looked at the numbers of sequences in the datasets. So that is the distribution before the contamination removal, and after the contamination removal, selecting the plant taxon as the target taxa, we can remove all the contamination with ContFree-NGS. And that is the run usage and time consuming of ContFree-NGS. And as you can see here, the wall time scales fast with the number of the reads, but the memory is constant. And as I mentioned earlier, ContFree-NGS is the first software that I developed. So I tested various ways to do this process faster. And here you can see the comparison of the versions 1, 2, 3, and 4. And that is the runtime and run usage for filter 10,000 reads. And as you can see here, in the version 1 and the version 2, it costs 11 minutes to filter 10,000 reads. And in the versions 3 and the versions 4, we have only 4 and 3 minutes. And that's because in these two versions, the version 1 and the version 2, I stored the taxonomic identification and the read identification in a Python dictionary, and this Python dictionary was very large, and this is very time consuming to do this removal. And with the SQL database, this process this process is a lot faster. And the same happens when I tried to filter 50,000 reads. So here we can see the time consuming and the run usage for the version 1, 2, and 3 and 4. And for the conclusions, ContFree-NGS is a very simple filter and useful tool that removes sequences from contaminating organisms in NGS datasets. Memory usage is low and independent of the number of classified sequences, and the wall time scales rapidly with the number of classified sequences. And as ContFree-NGS exploits the results from a taxonomic assignment engine, users must use the proper suite to achieve an accurate taxonomic classification. And that's all. Thanks, and feel free to ask if you have any questions or suggestions.
Thank you so much, Felipe, for your talk. It was very nice. I have also some comments in the end. So the audience can keep sending us questions as well. And now we have again Breno. Let's try again, Breno, to share your presentation. After we have Nelly. Nelly's also arrived. The audience is also congratulating Felipe. It was really a nice talk as well. Okay, let's see here.
Okay.
Oh, it seems much better now. Okay,
it's okay now.
Yeah, it seems so.
Okay.
Okay, so good presentation.
So we are just before 12 p.m. So good afternoon. I would like to present the paper featuring parts analysis of non-coding DNA/RNA sequences based on machine learning approaches. And the authors are composed by me, Breno Olívio. I am a Bachelor of Science Computer Science student at ICMC from University of São Paulo. We have also Álvaro Queiroz, Anderson Santos, Robson Buendia, Danilo Sanches, and my scientific initiation supervisor, André de Carvalho. So for the presentation, we have the following outline. Firstly, I will introduce the background behind areas linked to the feature extraction of biological sequences in bioinformatics. Soon after, I will show the methodology applied for our research, considering important tools for building our pipeline for the machine learning classification task. And more importantly, I will show the results considering our general pipeline created for tackling 2 case studies. And concluding the presentation with relevant assumptions considering the results obtained. Let's then begin with a context behind the errors linked to the idea of feature extraction of biological sequences. So over the years, with the accelerated advance of biological studies, non-coding sequences have attracted attention in bioinformatics. In elucidating the function of these non-coding regions is yet a key challenge, which has been addressed by several machine learning studies. Nevertheless, Machine learning studies applied to biological data require the extraction of features to identify patterns that allow their classification among other sequences. So, machine learning models need to understand in a relatively concrete way the characteristics of biological sequences, such as their statistics and other relevant information. Thereby, the feature extraction plays an important crucial step in machine learning. And there are many methods which are available in the literature for the idea of the extraction of biological sequences, such as the conventional feature descriptors. We have the well-known open reading frame and KMR. However, we also have other relevant approaches with mathematical descriptors. We have the idea of Fourier transform, entropy, complex networks, the representation in graphs, and they also could be considered for classification alongside conventional descriptors. Therefore, there's increasing demand for interpretable computation methods involving the understanding of the best features. In this study, we propose a pipeline using hybrid feature vectors. So the mathematical, which actually are little covered in encoding RNA, and the conventional descriptors. And to generate this hybrid vector, we use the math feature package with feature selection techniques and dimensionality reduction in our two case studies. So the first one would be the prediction of sRNA in bacteria, and we have the second case study, which is the prediction of circular RNA in humans. Thereby, we assume the following problematic. So, based on a hybrid feature vector with mathematical and conventional descriptors, which are the most important features for classifying sRNA and circRNA sequences? And our experimental results reported two contributions. So we have a high predictive performance in both case studies, and we have the second idea of the relevance of using the hybrid combination of mathematical and conventional descriptors. Let's then understand the methodology applied, which creates the general pipeline for our machine learning classification task. As aforementioned, we investigated different feature descriptors and applied in two domain problems, such as the prediction of sRNA in bacteria and the prediction of circRNA in humans. We propose a general pipeline considering our benchmarks for the predictive task analysis. And moreover, we evaluated a vector of hybrid features, so the mathematical and conventional, and different machine learning models behaving distinct organisms, in this our case, prokaryotes and eukaryotes. So for the prokaryotes, the bacteria, firstly we described the pipeline for this classification task, where we have created sRNA sequences from Barman and others from 2017, which also proposed an improved method for classifying sRNA sequences. And the data used are experimentally validated sRNA of Salmonella typhimurium LT2 or SLT2, collected by using the RFAM database. And our reference created negative data sets using the Emboss suite, the software shuffle-seq from this set of tools. And the same shuffle sequences are also used for the idea of reproducibility and the barrier comparison. In total, for this case study, we have used 182 sRNA versus 182 non-sRNA, which are the shuffled sequences for this pipeline. In addition, we have applied the math feature framework for a feature extraction package. And for the prediction of sRNA, we've used the following descriptors. So complex networks, channel intolerance entropy, open reframe, thicket score, and the dinucleotide and trinucleotide composition, the frequencies of this nucleotide. So we have evaluated the influence degree of each feature with the concept of mutual information, selecting features with most information gain. Mainly, we have used support vector machines, but we also induced random forest and XGBoost classifiers, but both had worse performance considering classification metrics. The Optuna library was used to find the most adequate parameters for the classification models, which uses a Bayesian optimization algorithm, and thereby 10,000 trials were performed with Optuna for each case, fixing the support vector machine's kernel as the radial basis function, and varying the regularization parameter and the kernel coefficient. Finally, we also included dimensionality reduction analysis. And the dimensionality reduction analysis is known for being used with linear techniques, such as the principal component analysis, PCA, but they cannot handle complex nonlinear data properly. Consider this, we have applied recent state-of-art techniques as uniform manifold approximation projection, the UMAP, for representing and visualizing the data. Non-linear techniques and dimensionality reduction techniques are being more recognized and can avoid overcrowding of the representation. For our second case study with eukaryote humans, we used the benchmark dataset provided by Bonidi and others from 2021. For the prediction, we have circRNA versus long non-coding RNA with in total 11,995 sequences, being 6,995 for circRNA and 5,000 for long non-coding RNA. And different from the previous case study, which was being compared with another reference, we have used AutoML, which uses the concept of meta-learning for finding the most suitable machine learning model for our problem and its hyperparameters. And also, considering a relatively large sample of biological sequences, we can divide your sample in a way to validate results beyond just analyzing the average metrics for, as example, the 10-fold cross-validation as we do in our first case study. In addition, we also use the math feature framework and structure features, descriptors such as the nucleic acid composition, GNC, and more others. We also evaluated the influence degree of each feature in this case study, but in this case using the gene importance as a reference index to determine the optimal feature subset. And to select the best classifiers, have used the AutoML, where all the steps are performed automatically using the idea of Bayesian optimization method, assessing, in this case, H2 combination of classifiers. With the best performance for this case, we has the ensemble learning technique, ADA boost combined with decision tree. So with our methodology and pipeline created, which were our results? And for the first case study, our experimental results show that mathematical descriptors such as complex networks, channel, and solid entropy can contribute to a certain classification with the conventional ones, like open reframe, thicket score, and dunucleotide and trinucleotide composition. And we realized that the TNC descriptor is a suitable descriptor for the classification, as our reference already did. However, achieved a considerable improvement when combining different descriptors. And it's important to add that shame shuffle sequences were used for all training testing, considering how the model performance can be influenced by how the sequences are shuffled. So it's important to add. And the ratio for the training testing was 1:1, and the results shown are the average tenfold cross-validation for each metric. And these are the results for our case with the support vector machines classifier. And we obtained the best average accuracy, average F1 score using the features with most information gain with mutual information, also using Optuna with the intent of finding the adequate number of features to improve the classification, specifically maximizing the average accuracy. And when we selected the features with the most information gain, we have the following proportion used for descriptors. And we can observe conventional descriptors had other variables used, but mathematical descriptors such as the solids and complex networks, even if they didn't have other features used, they had a reasonable number of features to contribute to our classification. Furthermore, features based on complex networks have an important highlight in terms of information gain. We can see, for example, in the first 5 features in this image are features from the complex networks descriptor, showing its contribution for an improvement for our machine learning classification task. And in addition, posteriorly to feature importance analysis, we apply PCA for dimensional reduction, and the data set was reduced to 100 principal components, maintaining the semi-classification performance. However, data visualization with PCA was not found to be a good representation of the data, since it showed notable variance beyond the first 3 components. Thus, for visualization, we adopted UMAP, which can create a better representation considering the possible observed non-linearity. And these non-linear techniques are becoming essential to represent non-linear complex biological data, although one of its weakness is the lack of strong interpretability of the PCA, as the dimensions of UMAP embedding space have no specific meaning. So because of this, did posteriorly. And finally, for the classification task, our results suggest that hybrid feature vectors, both mathematical and conventional descriptors, can give us a better classification for performance in terms of metrics as accuracy, F1 score, and AUC, area under curve, when compared to studies in the same benchmark data sets as a reference, which actually applied a single descriptor, the true nucleotide composition. And that is, using only this feature descriptor, we reached an average accuracy of 87%, which actually is 1.92% less than the vector with the top features. And for our second case study, so the select classifier, which was the ADA boost combined with decision tree, performed well in the classification task with robust results, both in training testing. And moreover, we realized that the feature extraction step contributes positively to the definition of the encoding RNA classes, with all metrics close to 95%. And our pipeline reached a low rate of false positives. And based on this, we investigated which features are most important to classify circular RNA and long no-coding RNA. And that case, for this case study, and we have this figure, which provides the best 10 features, calculated through the gene importance in a normalized way. And the Fickett score is the most important descriptor in the model decision, obtaining a very expressive value compared to the other features, which are in the top 10. And we also note that the two features tracked by the FICT score descriptor are present among this, the most important, reinforcing that this representation is very determinant for the circRNA problem. And we also elaborated this table with the importance score by the descriptor. And although the TNC descriptor is the most important in the journal group, it generates a large number of features, which is 64, while the FICT score generates only 2. Therefore, this information can be investigated by biologists to try to understand the meaning of these combinations of nitrogenous bases, and how can they be used to differentiate circular and low non-coding RNA. And finally, the importance of the features tracked by TNC descriptor represents about 37.7% of the total score. However, we emphasize that the division of importance between other descriptors as the FICT score and complex networks are extremely significant, again indicating the relevance of our proposal with hybrid feature vectors. Finally, with the results obtained, we have the following conclusions. In this paper, we have proposed a general pipeline to deal with different domains. So the prediction of sRNA in bacteria and the prediction of circRNA in humans. And for that reason, we have proposed different machine learning approaches with efficient hyperparameters optimization and also feature extraction techniques based on the MEF feature package. And to experimentally assess the best features, we have used 2 case studies. And the experiments, we obtained high predictive performance in both case studies. So highlighting the hybrid combination of mathematical with descriptors such as the entropy and complex networks. And we have also the conventional ones, such as the TNC. And finally, we realized the inclusion of hyperparameters, optimization, and AutoML improved the performance of non-coding sequences predictive models. So now concluding the presentation, first of all, I would like to thank the organizer of the symposium and the opportunity to share this research and others, contributing to national research development with knowledge sharing. And the authors would like to also to thank CMC USP, VTPR, CAPES, and São Paulo Research Foundation. So thank you very much for your attention.
Hi, everyone.
And please, if you have any questions or would you like to contact me, this is my email. So this is it. Thanks.
You are muted.
I'm sorry. So I thank Breno for his talk. It was really nice. Thank you, Renato. Renato is also saying me my microphone is muted. So I'm calling Nelly again. He's going to present his paper. It's a short paper entitled CEvADA Coevolution Analysis Data Archive. So let's listen to Nelly's presentation, and after that, we are going to have some questions here. So welcome, Nelly, and can you share your screen too?
Okay. Thank you, Raquel. Can you see my screen?
Yes.
Okay.
Okay, so go ahead.
Hello, my name is Nelly, and I'm going to present the Coevolution Analysis Data Archive, CEvADA. So this is a short presentation. I will be brief in my talk. Multiple sequence alignments are one of the main data formats in computational biology. We can extract many patterns from them, such neutral selection positions, low or non-selective pressure, and can handle mutations without large effects in the protein function or stability. In contrast, we have the highly conserved positions. It gives a strong indicative of evolutionary constraint, and usually mutations in these positions tends to affect the protein function or stability. Sometimes the conserved position are not so obviously, because the selective pressure is not on a specific amino acid, but on a stereochemical or structural property. One example is the hydrophobic residues. They are usually conserved in the nucleus of the protein, since the substitution in that region might cause a large impact on the protein structure. So biological process like gene duplication followed by divergence can lead to the emergence of new proteins functions, although they descend from the same ancestor. Therefore, these are patterns that can also be mapped from a multiple sequence alignment comprising a superfamily. In this case, one of the ways to detect superfamily specific feature is by looking to the local conserved positions. It happens when the amino acid or the stereochemical property is conserved only in a subset of these sequences. One efficient approach to find the local conserved residues without need of the pre-classification of the sequences is using covariation analysis by measuring the tendency of co-occurrence in pairs of the residues in the alignment. Therefore, if at least two residues is locally conserved, they will be catched by measuring the tendency of co-occurrence of these residues. Due to these characteristics to find be related to specificities in alignment, covariation analysis can be applied in many problems like protein annotation, mutation effects, prediction, determination of secondary structure, orthologous identification, protein characterization, functional residues prediction, and prediction of active and allosteric sites. And when we link each pair of residues by their tendency of co-occurrence, we have what's called the covariation networks, the amino acid covariation networks. So in this project, we developed a database of covariation patterns in protein families. This is the home page. You can navigate through the family, search by a PFAN ID or a UniProt ID, or even a sequence here using the Hammer Search API. And you can also find the documentation about how to use our REST API to perform programmatic access. CEvADA was built under the PFAN architecture, so we're using the same architecture of PFAN with a mirror of the database, and using the Conan standalone software to calculate the covariation. So to be very briefly in the pipeline, we first apply a set of filters to remove the redundant sequences and fragments. We then convert the multiple sequence alignment into a bipartite network of sequences and the residues. We make the projection of the residues that occur in the alignment. We then set a p-value to retain only the statistically relevant edges to finally perform a community detection algorithm to classify the clusters of covariating residues. At the moment, we are covering 35% of PFAN, including the alignments that has at least 500 non-redundant sequences and a maximum of 20,000 sequences. We have more than 45 million sequences in our database and 6,000 protein families. this side of the non-covered protein families are hard to us because they have a very a tiny set of non-redundant sequences, and since our method is based on statistical tests, it will not have the good confidence to calculate the patterns from here. But this side we are still calculating, and we are planning to release in the next versions of the database. So we have two types of entry pages, one based on the protein family and another based on the sequence. Here I'm showing an example of a protein family page. The page is, in this example, the page is extensive, so I will split in a few slides. So it contains the descriptions of the domain. This is automatically fetched by using the Wikipedia API. Then we have an option to change the cutoff of the network, another option to download the raw data, and the interactive table to see the sequences that are members of this protein family. And we also have links to access the CEvADA page. Then we show a graphical visualization of the covariation network, colored by the clusters, followed by the location of the detected residues in a consensus sequences, and a table listing the detected residues and the clusters. To finally, we show the covariation matrix for each cluster. Therefore, we can see that the residues detected in the comminute 1 are conserved in around 30% of the alignment, and the residues detected in comminute 3 are conserved only 5% of the sequence in the alignment, and comminute 5 is highly conserved on the entire alignment. So we can detect the overlapping local conservation without predefinition of this subset. In our group, we have been working with this subject for a while, and we have seen that many users of our computational tools are not actually interested in studying the evolutionary history of a protein family, but they are interested only in key features of a particular protein. So this one is the main reason that why we designed CEvADA, to provide a protein-specific evolutionary information. For that, we also provide sequence space. Here you can see some basic information about the protein. These are collected from the UniProt PFAN. We also show the location of functional features like disorder, region, low complex, synopeptide, transmembrane, and the PFAN domains that are mapped to CEvADA with link to them, to the page. And we also show the detected clusters of correlated residues for each PFAN domain that are available in CEvADA. So matches are displayed in green, while mismatched are displayed in red. So we can see that this protein have all the residues detected in the community 3, but none of the residues detected in community 4. And finally, we provide 2 REST API endpoints for fetching our data by programmatic access. You can find data related to a protein family or related to a protein sequence. And you get everything, all the results mapped to the position of the specific sequence that you want, or mapped to all the sequence from the alignment. So I would like to thank the support agencies, the team. I also would like to thank you to Professor Miguel for technical support, and I'm open to questions now. Thank you.
Thank you, Nelly. Excellent presentation. I'm going now to call all the other presenters here. All right. Okay, so let's start. I ask the public to write down the questions in the chat here on the YouTube channel. Myself, I have several comments and in questions as well. But I would like to start with a question from Marcelo Reis, from Instituto Butantan, for Gabriel. He says, Gabriel, congrats for your talk. Your preprocessing resembles the application of morphological filters. Did you consider to benchmark preprocessing plus CNN against deep morphological networks?
Thank you for the question. We didn't make this kind of deep networks, but I think it's important. I think that it can help to achieve better results compared to our pre-processing techniques. And I think in the future, in our future experiments, can be considered as an experiment to apply to our model.
Okay, thank you, Gabriel. Well, I have several comments here regarding your talk, but just a curiosity here. How did you do that heat map analysis that you presented with the images? What tool did you use? I thought it was very very insightful, in fact.
We used GradCam. It's a common tool for this kind of analysis, and it uses the last convolutional filters of the network to see what the network is paying attention on the image. So it's very interesting to understand
Yeah.
the
Yeah.
decision.
In fact, I was ready to ask you if it wasn't expected that the pre-processing was not necessary because of you were going to use the convolutional neural networks. They deal well with the noise and things like that. But after you showed that it was very nice with the heat maps because you just presented it visually, so it was very very nice actually. So last question from myself. And I don't know if there are others here from the audience. If there are, I can also deliver. But why did you choose these two architectures? Do, can you explain briefly why these two specific architectures?
We choose the first one, the baseline, because it's very simple and it can run faster. So we can make experiments with different preprocessing techniques in a faster way than the ResNet, for example. And the second architecture that we used was ResNet 50. This architecture achieved good results in many medical image processing and analysis diagnostics. So we choose these two architectures for these reasons.
Okay, thank you.
Thank
And
you.
let's see here my notes. So for Felipe, I was very happy to see your presentation. It was very nice, especially if I'm not mistaken. You said you are undergrad students, right?
Yes.
You are from biotechnology, yeah. I
Yes,
think
yes,
so.
yes. From
So
biotech.
it was
Biotech.
very nice your work. I really like it, and I have some questions. One is why you mentioned that you use the SQL to improve the performance, right?
Hmm.
Do
Yes.
you think you could improve this performance, improving your algorithms, improving your methods, and maybe the data structures you use or things like that? Just a curiosity, okay?
Yeah, yes, of course, it can be improved. As I said, that's my first software, and I tried a lot of ways to build that. And this SQL database was built with a BioPython function. So I don't know. This can be proved, but it's out of my knowledge
Yeah,
right now
yeah.
in this moment.
Yeah, no problem. It's just because it was noteworthy that you really pursued this question, this improvement of the software. It was nice
Hmm.
to see the experiments you showed us with the performance and the use of memory and things like that. So you really, I think that you really went farther. Um Considering your background, your initial background. So
Hmm.
it was really nice to see, and I think that you certainly are going to obtain this knowledge you were mentioning that it's a little bit far from your background. So
Hmm.
it was really very nice. Congratulations
Thank
for
you.
this, and also the public left here several congratulations for all of you, all the presenters. It was really a nice session. Um, for Breno, I took some notes as well. Um, what I missed here in your presentation was I was wondering because you mentioned you are your background is computer science, right? So it's the other side. Uh, I was wondering if you didn't you want to go further to better understand the biological meaning of your findings? I mean, you mentioned that TNC was much better. You also mentioned that the authority score in the complex networks was nice as well, was important. And I always I always want to go to the biological meaning of these features. And I know your background is computer science. So what do you think? Have you discussed a little about with your collaborators? And what can you tell us about this biological meaning? How do you intend to address this?
And absolutely, I want to, as future works, I want to go further in terms of understanding the meaning actually for these scriptures. Um, but with these results for this paper, it's already how can I say an initial step for a bigger perspective where when I can select some specific things and I'll try to tackle the and understand better these descriptors. But absolutely, I want to understand even further.
Yeah. That's not a criticism, but that's just because we are here discussing science and the future perspectives and so on. So it was really something that I missed here in the case of your presentation. And you presented also very nice 3D maps of the sRNAs and non-sRNAs, right? So another question that I have is why do you think that the cluster you had two clusters? One, I think that in the UMAP part, you have one cluster of the sRNAs and the other with the non-sRNAs. Why the non-sRNAs are much homogeneous? Do you have you noticed that? I was expecting that the sRNAs were more, you know what I mean, were more homogeneous, and the others were more sparse, and it was the opposite. I thought it was curious.
It's an excellent observation. I- Considering the question that these non-sRNA are shuffle sequences, I think it's one of the things to take in consideration. And these shuffle sequences have some random characteristics in them. So there's this, and it happened. But this is something to study even more, to deal with shuffle sequences, because it's interesting way to classify in the models. In our second case study, we actually used real biological sequences, which was the long no code and circRNA. But for this first one, this case it was really particular, I could say.
Yeah.
So, but really interesting what
I think
happened.
so. Yeah, I think so. Okay, thank you, Breno. And we have here a question from the audience. Professor Valdeir says, he says a question for Nelly. Could you just clarify the threshold used for the maximal identity? Is there a particular reason for your choice, which is maybe related to this protein group? I had this question as well. Go ahead.
Okay, so this is to remove evolutionary bias. Imagine that you have a subset, a sequence alignment with 90% of the sequence are from the same group of proteins, a sequence with identity higher than 90% of the sequence are coronavirus sequence for different strains, and the rest of the sequence, the other 10%, are the sequence from other viruses from the same family. So what we will see in your alignment is that everything is conserved, but they are not real conserved. It's just a evolutionary bias in your alignment. So you need to remove this redundancy before do any statistical analysis. This is the same that is used in phylogenetics. Most of the times when we're doing statistical analysis on multiple sequence alignment, you need to treat this bias.
Right. And I would like to listen from you a little about this regarding this reducing of the redundancy. And you mentioned briefly, because of the lack of time, of course. You mentioned the bipartite graphs, and I thought it seems very interesting. Can you tell us a little bit more about it?
Yeah, this is just a way to see the alignment as a graph. So the way that we treat the multiple sequencing alignment, it's like a bipartite graph where we have a set of nodes that represent each residue in the alignment, and we also include stereochemical properties as residues. And in the other set, we treat the labels of the alignments. So when you make the projection of this graph, you can either have a projected network that contains only residual nodes that are linked according to the co-occurrence in the alignment, or label nodes, that is proteins that are linked to basically identity, to have the same sets of residues. And when you do, we pass a p-value and do a thresholding using this p-value, you can remove the redundancy, and then we'll have a signal of tendency of co-occurrence in the alignment. It's a basic pipeline that it's applied in most of bipartite networks analysis. It's using on gene disease, on social networks. It's a general pipeline for dealing with tendency of co-occurrence in networks.
Right, very nice. So let me see. I don't think we have more questions from the public, but we can, if you have some questions, please write it down in the chat. I would like to thank all the presenters. I think that all the presentations were very clear, very interesting. and you all had presented very high quality works that I really appreciated. And just to recall everybody that we have today at 2 p.m. a special session, Bioinformatics and Artificial Intelligence. The chair is Dr. Maribel Hernandes Rosales. So it's going to be very interesting, several presentations and discussions about this hot topic involving artificial intelligence and computational biology. So we wait for you there at 2 p.m. And later we are going to have a meeting of the Special Committee of Computational Biology, which is a committee from the Brazilian Computer Society that organizes this event. So everybody is invited to participate. One that was enrolled in BSB. So we are going to present some statistics of the event and discuss the next action. So we are all invited. Okay, I don't think we are going to have more questions. So I finished this session. Thank you all again, and thank you for your presentations, and see you in the afternoon. Okay, thank you.
Thank you, Raquel.
Thank you, Raquel. Thank you, everyone.
Thank
Bye
you.
Thank you, everyone.
Bye.